Yes, Raj.
Yes.
Okay, fantastic.
Good morning.
Good morning. Okay, we'll try and, it's a slightly long class, which we'll try and finish slightly quickly because there's a seminar on as well. So let's just get into it. Okay. Great. So today we're going to talk about cell behaviors. So this is one way to think about cells so that we can start thinking about tissues, and we can start thinking about how elaborate morphogenetic movements, so changes in the configuration of cells and the shapes of tissues, in the differentiation of structures occurs in the developing embryo. And so this is the chick embryo here, and you can see that as it develops, it transforms from a kind of disc of tissue into this fairly beautiful and complex structure. You can see that the embryo is elongating, that there's something going on here, which we'll talk about a little bit later. Here the embryo is elongating, which we'll talk about later as well. It's becoming narrower, which again we'll talk about later. Here you can see the neural tube, which will form the central nervous system, folding and becoming a tube. You can see here the heart starting to form here. So all of these behaviors are being exhibited by individual cells, but they're all being coordinated so that no one cell is doing something that another cell doesn't know about. Okay, so we've already talked about one cell behavior, which is proliferation. Today I want to talk about a few more cell behaviors, but I want to think first about what kind of subcellular structures could be controlling cell behavior. So let's, I'm sure all of you remember this. This is, and I'm saying remember because I think this is basically intermediate school stuff, elementary school stuff. So we have a cell. The cell consists of various organelles, including, of course, the nucleus, Golgi, mitochondria, and these other structures, such as the microfilaments, microtubules, and intermediate filaments, which make up the cytoskeleton. And I'm going to talk a little bit about the cytoskeleton as a way of introducing some of the behaviors I want to talk about. So the plan for the lecture is to talk about the cytoskeleton, think about how that could be directing some cell behaviors, and then bring in an example of a coordinated bunch of cell behaviors that need to happen for one particularly fundamental process in development, which is gastrulation. Okay, cool. So the cytoskeleton is here. So let me know, so I think everyone knows about the cytoskeleton, is that correct?
Yes.
Yes? Great. I'm gonna try and get a mouse. Can this work? No. Sorry, I'm just trying to see if I can get some kind of pointer or something like that. Spotlight. Does this work? No. Okay, I can't do it. And so,
Raj, I think if you right click you'll get to have one pointer.
Really? Who
I
said
guess
that?
so.
Who said that?
Nivedita.
Thank you. Thank you. Right click. I'm on a laptop, so I don't even have a right click. Shift and click? No. Control click? No. Option click? No. Command click? No. No. Okay. All right. So never mind. Never mind. I'm kind of giving up. Okay. So no one sees this kind of format tab that's been opened on Zoom, do they? No? You're just okay. Okay, good. All right. So what you're looking at here is various structures in the cytoskeleton. There's of course microtubules. Microtubules are made of tubulin, tubulin dimers. Actin is made of actin monomers, and then you have intermediate filaments. Here, we're thinking about intermediate filaments that are made of keratin, but intermediate filaments can be made of other things such as vinculin as well. We're not going to talk so much about intermediate filaments for the rest of this lecture, just because not much is known about them, and their dynamic control doesn't seem to play too much of a role in the embryo. So, like I said, we have these three main types of cytoskeletal polymers, and they're all polymers: actin, microtubules, and then this other group of polymers known as intermediate filaments. So together, these control cell shape and cell mechanics. And so they're organized in networks. They're there because they're somehow resistant to deformation. So you can stretch and squeeze a cell and it won't change shape too much. It will, of course, eventually, depending on how much stress you put on, it will eventually change shape. But these act as actual, the word skeleton is there for a reason. They actually act as the skeleton of the cell. The polymerization and depolymerization of actin filaments and microtubules kind of also generate directed forces within the cell. They drive changes in cell shape, and they have motors that kind of transport cargo either on microtubule tracks, as you can see here in B. They allow actin filaments to slide over each other, as you can see in DE here. Okay, so the actin filaments sliding over each other is how muscles contract, for example. It's almost an identical process. In the cell, actin filaments slide over each other with the help of myosin motors, as you can see here in C. And this causes some level of contraction, even within the cell. This contraction is important for changing the shape of the cell, and we'll go into that in a little bit more detail in the next few slides. So I was going to kind of lecture on the differences between microtubules and actin filaments, but I think many of you have done this before. So I'm going to ask someone to tell me what the main difference between microtubule and actin filaments are. Anyone? Can you hear me?
Yes.
Okay. Okay, so if there's no answer. Okay, so the main difference is that actin filaments are Let's see if I can stop sharing. Okay, so I'll try again. So the main difference is that microtubule filaments are much more stiffer than actin filaments. And so this causes, so they're kind of persistent as well. They have this kind of self-organizing activity. So they'll ramify through the cytoplasm from what's called the microtubule organizing center, which is generally sat close to the nucleus, to the edge of the cell. And these kind of stiff tracks are then used to transport vesicles, transport organelles, and actually fix organelles in position. Actin filaments are much more dynamic, and so they, as I mentioned, can be contracted, they can disassemble and assemble very easily, and they also form branch structures as well. And so these branch structures, and I'm sure once you all start cell biology next semester, you'll go into this in much more detail. But these branch structures are formed, they're important because then you can start exploring a range of actin structures that enable various processes to occur. And so one process is, of course, in the formation of a neuron, for example. Neurons form, you have a cell body on one end. Here you can see that in A, with a dendrite, with an axon, going out into a growth cone. And the growth cone is exploring and looking to make connections with other growth cones or target tissues that this neuron will want to innervate. So the growth cone, if you look on B, consists of actin in red, and in green it consists of microtubules. And you can see that the growth cone is busy ramifying through the organism, looking for various structures. It's sending out these filopodia and lamellipodia and exploring its space. When it doesn't find a particular its target tissue, you get retraction, and this kind of dynamic turnover of actin is important for this activity. You can also see that some of the actin shown in D here in this cartoon is branched, and this branching happens because there are accessory proteins to the actin filaments which can set up new branches and new kind of morphologies of the actin filament. So, for example, in the growth cone, you can initiate actin. So here in A, you're looking at the edge of a growth cone. The red is actin. The green, I think, is microtubule, but I'm not. Yeah, it's microtubules. No, this is not a growth cone. This is a fish keratinocyte. But it's the same thing. What you're seeing is that you have a signal. So if you look at B, you have a signal which causes the recruitment of an adapter. The adapter recruits what's called in this review a constructor, and the constructor is something that initiates the actin filament. The actin filament elongates, and as it elongates, other constructors can bind to various branch points in the actin filament to make different branches. And what you're looking at in C is kind of this happening in vitro. And so by adding particular proteins, this actin branching structure can be recreated in vitro without a cell, and just based on the individual components that make these structures. So this is one way that by modifying the actin cytoskeleton, you can get very dynamic and fluid changes in the shape of the cell. But what we're trying to think about is how then this influences cell behavior. So I'm going to come back to these kind of points in a little bit. I'm putting this up. I want you to, if you can tell me, you're all aware that cells can either be epithelial or mesenchymal. Is this the first time you're hearing this, or you've heard this before? Yeah, sorry?
I've
Am I
heard
audible?
this, but I don't have a lot of clarification regarding these terms.
Okay, fantastic.
I
I'm
have heard about this too.
okay. Okay. So then I'll go into this in a tiny amount of detail. Sorry. It really is very difficult without a mouse. Okay. I'll go into this with into a tiny amount of detail. So cells can be either epithelial or mesenchymal, and they're just two different morphologies of cells. It's more complicated than that, and we'll go into this in a
Raj,
little while. Yes.
if you don't keep the full screen view, then you can use the pointer, I guess. It was
Yeah.
visible.
Yeah. What would you want me to do? Do you need a pointer, or do you
I think a pointer would be better.
okay? All right. Thank you. Okay. All right then. So you can see all of this then, yeah? You can see slide only. Okay, there we go. So in A is, so you can see the pointer now, yeah? You can see an arrow?
Yeah.
Yeah, good. Finally. So where was I? Epithelia. Okay. So what you're looking at in A is an epithelial cell, and in B is a mesenchymal cell. The first thing you see is that the epithelial cell is kind of has neighbors. So epithelia usually are not usually, they're never solitary. They're with other, they form a sheet, and these sheets have connections between each neighboring cell, and these connections here, they're marked as apical, the apical junctional complex. The apical junctional complex contains adherens junctions, which I think you've all heard of, tight junctions. There are desmosomes further down. There are integrins at the bottom. Not integrins, sorry. There's attachments to the basal lamina at the bottom. So epithelial cells always sit on the basal lamina. They have an apical basal polarity, and so epithelial cells know what's up and what's down. And they have these networks of actin on the apical side of the cell. Okay, and so essentially when you talk about apical and basal sides of cells, what you're thinking about are epithelia. Mesenchymal cells do not have to be do not have to have neighbors. They can be quite solitary. They migrate, so they're migratory. They don't have a apical basal polarity. They can and they have this kind of organization of actin and myosin, so that actin is generally concentrated on the leading edge of the mesenchymal cell if it migrates, and the mesenchymal cell will make attachments to the substrate using integrins. Okay, we'll go into more detail about epithelial cells and mesenchymal cells in subsequent slides. Okay. Now I just need to be able to change the slide. So let's just talk about epithelia for a little while. There are various types of epithelia. The simplest epithelia are just a single sheet of epithelia. They can be squamous, cuboidal, or columnar based on the aspect ratio, so the width to length ratio. These kind of become important when we're thinking a little bit later about epithelial differentiation. But right now, we'll just stick with these kind of simple definitions of columnar epithelium. Columnar epithelium can also be pseudostratified. And let's think about stratified epithelia. So as you can see here, stratified epithelia are just simply multi-layered epithelia. You can have stratified squamous epithelia, stratified cuboidal, or stratified columnar epithelia. And these, so once an epithelium is stratified, it's generally fairly differentiated. So these kind of structures you see in the skin, for example, or in various parts of the intestine when we're thinking about stratified columnar epithelium. Pseudostratified epithelium is here. You can see this. This has a very particular kind of developmental potential. Generally, epithelial tissue which will give rise to various organs, so such as the neural tube, will form from pseudostratified epithelia. It's called pseudostratified because even though it's one sheet of epithelia, the nuclei are placed in different positions within this epithelium, giving it the appearance of having multi-layers, or even though it's only one tissue layer thick. I'll come back to this in a subsequent lecture because it's a pseudostratification and the mode of division in pseudostratified epithelia deserves kind of more attention than I'm able to give it right now. Okay. So, any questions so far? I haven't done this so much justice yet, but it will all come together soon. Okay. If there's no questions, then let me go back to thinking about epithelia and actin, and just tell you that epithelia can be reconfigured by actin-myosin activity. So, as I mentioned, epithelia form sheets. They are next to each other. They are connected to each other by these green junctions, this apical junctional complex. And this apical junctional complex is used to brace, to attach actin filaments onto it. These actin filaments are also decorated with myosin as well. And so because of this, these actin and myosin filaments, you start getting contraction that's attached to the apical junctions. And in this way, you can start changing the shape of the epithelia. So for example, here you can, by contracting either the apical cortex or the junctional actin, so there's a band of actin that runs on the junctions as well, you can start contracting the apex of the cell. So epithelia will go from this, basically this square shaped or this columnar shape to something like a wedge here, okay? And so once you're able to do that, you can see that an epithelial sheet can start exploring lots of different types of shapes. And so you can go from a flat sheet to a tube, for example, if you can control the way that the actin is is contracted, you can go either to a ball as well, to a sphere, and you can start forming ducts and tubes, all with epithelia. Okay. Any questions so far?
Yeah, Raj, I had a question.
Yeah.
Apical side and the basal side, are they fixed?
Yeah, fairly fixed. It's really hard for an apical side to become a basal side. And so I went into this very, very briefly, not even briefly, I didn't go into it at all. There are particular activities that are always going from, that are apically directed. So things like protein secretion will happen either apically or basolaterally. It will never happen. So a particular protein that needs to be secreted will either be secreted to the apex or to the base. It will never be secreted in both directions. You have...
I'm just confused because that picture shows kind of the opposite. The basal and the apical side is labeled.
Ah, here. This is, yeah. So here, I don't know what this is happening here. I've always... so, yeah, I don't know what's going on there. But it's always, this side is apical.
Okay.
Oh, I know, sorry, yeah, I do know what's going on. I think this is kind of a particular part of the embryo, which I'll talk about, or maybe Bhavna will talk about. It's kind of the neural tube lumen, where the configuration is slightly more ambiguous, and it kind of fits in with pseudo-stratified tissue as well. Okay, and we can go into that in another lecture. Maybe I'll put in a slide next lecture to talk about that. Okay.
Okay.
Okay. But changing shape. So we can change epithelial shape by kind of changing the way actin contracts. Okay. So let's have a look again at this developing embryo. And as I said, what you're seeing is something going on here, but also you're able to see the epithelia changing its length and narrowing as well. Okay, and we kind of want to understand how this is happening, and so in the next couple of slides, I'll try and explain this to you. Unless anyone has an idea how this kind of process could be happening. I'll give you a kind of clue, which is when we look at the formation of the Drosophila larvae. And so what you're looking at here are all the edges of the cells in the Drosophila larvae labeled up. And this is also marking this is marking actin in all the junctions of the cells. And when I run this movie, what will happen is you'll start seeing that the cells are changing shape, and it's somehow causing an elongation event in the embryo. So are you able to see this?
Yes, Raj.
Yeah.
Yes.
So I want you to tell me what you think is going on here. I mean, let's ignore these round structures here. Okay, this is cell division going on here, and we'll go into that in a little bit. But this other tissue, these other epithelial cells around seem to be doing something. And so is anyone able to describe exactly what they're doing?
Raj, Yojit here.
Hi, Yojit.
Good morning. Maybe the cells, the epithelial cells at the boundaries are secreting proteins or morphogens that are like forming a gradient of sorts, which is why these cells are then, the cells at the center are proliferating or getting into elongation process.
Yeah, yeah. So that's one thing. Yeah, yeah. So probably that's happening, but there is kind of this. It's maybe a little bit difficult to see, but there is a kind of a change in where epithelial cells are going. Okay, so if you focus on one epithelial cell,
They're kind of folding in.
yeah, kind of folding in, not quite. So I'll tell you that all the cells are not really dropping out; they're all staying in place. But you're seeing this kind of elongation event as well happening, yeah. And maybe it's not so clear here, but while all of this is going on, the animal is the fly maggot is kind of elongating as well.
Raj, is it possible that the cells are, I mean, basically the nuclei of the cells are dividing, but the cell is not dividing? So there's an elongation. I mean, there's like karyokinesis, but no cytokinesis. Is it
Yeah,
so?
so that's what. Yeah, so that happens in some things, but it's not happening here. This is so that's also a great guess as well, because that does contribute to this. But there is cell division, and so what's happening is that the epithelia is remodeling, and the way it's remodeling is by reorganizing. And so what we were seeing some random cell divisions here. So random cell divisions are adding more epithelia. So that epithelia needs to go somewhere, and that may cause the epithelium to get bigger. There could be oriented cell divisions, and so the cell division plane can always be restricted to one direction. And so when you do get cytokinesis, you're elongating the tissue as well. You can get cell competition as well. And so here, this is another way of thinking about things, that one population of cells may proliferate faster than another population, and that causes some buckling. If there's two populations here and here, this bottom population is proliferating faster, this population will be displaced, and you'll start getting various morphologies that reflect this. And the other thing that could be happening here is cell rearrangements. And I'm going to talk a little bit more about cell rearrangements because they're quite important and quite interesting. So remember, around every epithelial cell, there's a junction, and the junction is loaded with actin and myosin, and the junction is stuck to another cell. So if you want to move one cell to in place, you're going to have to somehow bring this cell. It will have to reorganize its junctions so that it has a new neighbor. And so the way this is happening is that junctions are being collapsed down. So a junction will come down, and then this junction will then move across this way as well. I'll show you in the next slide. I think I'm doing a terrible job of explaining this. So here, okay. So what's going on is you get what are called rosettes, and these rosettes are because a group of junctions, here we're seeing 1, 2, 3, 4, 5 cells. This group of junctions is collapsing down thanks to myosin activity to form a rosette. And so now you've gone from 5 cells all in a row to 5 cells forming a flower, all next to each other in a vertex. Now what you can do is now expand all of the junctions in the other way. So you're expanding the junction this way, and so you're resolving the junction. So you go from 5 cells that are facing north and south to 5 cells that are facing east and west. Okay, and this happens because of myosin activity, and we'll talk a little bit more about myosin activity in a subsequent slide. But I'll go back to this movie and let you then look for rosettes. Okay, and so there's lots of rosettes that you're able to see here. Here's one forming, and then you'll be able to see that resolving in the opposite direction, so that you start getting this kind of epithelial remodeling. So here's many rosettes are forming here and here and here. Okay, and so that's one way of kind of molding, playing around with epithelia that are in a sheet, so that you can start exploring different kinds of structures that will give rise to different organs. Okay, so the process you were looking at was germ band extension in the fly. Germ band extension, as I said, kind of causes this elongation of the maggot, so that this structure now loops around here, and this happens because of both cell shape change and this cell rearrangement. And the cell rearrangement is happening because of junctional collapse and junctional extension. Here, this is showing a group of 4 cells, which is easy to visualize. And here we're looking at a group of 7 cells, which are showing this very beautiful junctional remodeling to change the neighbor relationships of epithelial cells. Okay. Any questions here? I will, so if you're not quite getting it, we can just contact me, and we can sit down and explain and talk about this a little bit more. Usually when I'm lecturing this in person, I would get you all to demonstrate junctional remodeling. And so I would have you all stood up with hands, with arms linked, and ask you then to change your neighbors so that you can form different configurations. But you can all imagine how this could happen, okay? So please let me know if you have any questions. Good. And you can all see my screen, yes?
Yes.
Okay, thank you, thank you. Basha, if no one can hear me, if my connection is lost, please text me, okay?
Yeah, Raj.
Okay, thanks. Okay, so these behaviors kind of need to get coordinated, and in the next couple of slides, I'm going to talk about gastrulation. So if you can let me know if you know about gastrulation. Has anyone done gastrulation?
It is like a movement of cells which leads to the formation of the 3 germ layers. So that's basic that I know.
Okay, fantastic. That's almost completely what I'm going to tell you. Okay, so so you're right. So what we're going to talk about is the formation of the germ layers and gastrulation, and this is a kind of very nice example where different behaviors are coordinated to give a particular structure. So as Kanika says, it's forming, gastrulation is important for forming one of the germ layers, which is called mesoderm. And mesoderm, so all of you know the 3 germ layers, yeah?
Yes, Raj.
Okay.
Yes.
Yes. Okay. Ectoderm, mesoderm, endoderm. Yeah. Ectoderm is on the outside, mesoderm in the middle, endoderm at the bottom generally. Yeah. So most animals are what we call triploblasts, which means that they have 3 germ layers. And so the so up until the invention, so what you're looking at here is a evolutionary tree of animals and what are thought to be our closest relative, conoflagellates. Animals go from these from sponges, jellyfish, all the way insects here, and we're here in chordata. Okay, and there are a couple of big splits that have happened, and the first big split after the invention of animals that happened is the kind of evolution of bilateria. And so here bilateria have triploblastic organs, which include mesoderm. Okay, and so all of these animals will have mesoderm. Jellyfish and hydra have what are called mesoblasts, so they have an internal structure which does not arise from gastrulation, but probably has some hint of gastrulation there, and we'll go into that in a much later slide, much later lecture. So the germ layers essentially are ectoderm, mesoderm, endoderm. Sorry, luckily this is not a slide view, so I can do real-time corrections. Ectoderm gives you epithelial tissue, basically, the skin, brain, the peripheral nervous system. Endoderm also gives you epithelial tissue, gut, pancreas, lungs, pharynx. Mesoderm will form all of these structures as well, okay? And this is a much more complicated germ layer flowchart that I've taken from Carlson, showing all of the various structures of all the derivatives of all the various embryonic layers, okay? And we don't need to go into that in too much detail. But what is important is that mesoderm is in the middle of all the tissues, but it's the last one to form. So somehow the embryo needs to get mesoderm from somewhere outside to somewhere inside, and it does that through gastrulation. And so these are all different animals that are gastrulating. What you're looking at here is the sea urchin, and the sea urchin gastrulates like this. These blue cells here are going to be mesodermal. They'll fall inside the cavity of the sea urchin embryo, and then they'll start to direct invagination, so shape change of the endoderm, and they'll end up inside the embryo. Here we're looking at this next panel here. We're looking at Drosophila. Again, the mesoderm is induced in this structure here. It will invaginate. So here, this is a simple apical constriction of the mesodermal cell, and this will then constrict so much that it will go inside the embryo and then change its shape so that it becomes mesodermal like this. Okay, so these green cells are mesoderm, and in the Xenopus embryo, you have a more complicated gastrulation process, which I'll go into in the subsequent slides. So this is sea urchin, and this is Xenopus. so what happens in Xenopus is the at some point at the border of the ectoderm and endoderm cells, you have this pinching of the dorsal lip of the blastopore. So I think I've introduced the dorsal lip once before. Just to check you're paying attention, can you remember where you've heard dorsal lip in previous lectures?
Organizers and the role of organizers and the transplantation experiments of.
Fantastic, fantastic. I'm so happy that one of you remembered. I'm sure all of you remembered, okay? But thank you, Shubha. Yeah, so the dorsal lip is, in Xenopus will form was famous for the organizer experiment. And so this is the first tissue that undergoes gastrulation, that starts to invaginate. So these cells, again, will form a wedge shape, and this wedge shape starts driving in invagination of mesoderm. And I'll show you how it drives invagination in a few slides. This tissue here, this red tissue, which is now the mesoderm, will start crawling along the roof of the blastula. So this is called the archentron. It will just start crawling here, aided by this kind of the blastopore lip, until finally, so this mesoderm will crawl all the way around and will form a layer of mesoderm in the embryo. Okay, so let me explain how this is working. And so this is a view of gastrulation happening. You can see here now the crawling along the mesoderm along the top of the archentron. Here we're going in reverse again, and so this is gastrulation in amphibians. So you do get some crawling the other way as well. So you get the ventral mesoderm joining as well at some point. Okay, this kind of process is driven again by epithelial remodeling, and so this is shown much more clearly in zebrafish as well. Zebrafish gastrulate in a very similar fashion. What happens is that this ectodermal tissue is epithelial. It remodels so that you get a general thinning of the epithelium, and as it thins, this epithelium is starting to be driven down, and as it's driven down, because you now have a kind of what's called a bottle cell on one side, that bottle cell then starts driving the tissue the other way as well. This is shown here in zebrafish. You get these blue cells here rearranging. You get the cells, the bottle cells, kind of driving cells inside. So as these top cells kind of start to thin and push down, other cells on the edge are being driven inside because that's the only place for the force to go. And so this is kind of the way that this has been explained to me is it's kind of like putting one of those beanie hats on, yeah. And so you pull the hat down like this, and this is the same thing that's happening here in a pipili. Okay, you can probably see this much more clearly in this movie, and so you can see here. Let's look at this view, and you can start seeing now the cells starting to undergo a pipili here, and now you can see them here, and then they'll start to invaginate and then form the embryo. Okay. Okay. Okay, any questions here? So there's some very nice reviews about this, especially from Marita, who talks about this from a physics viewpoint as well. And so they talk about this this movement as being a kind of active fluid model with different types of surface tension, which enable these kind of movements to take place. So I will put up these references as well, so that you start getting a better appreciation of what's going on in these kind of different gastrulation schemes for these for xenopus and zebrafish. But the main point of this, and I will go into gastrulation in later lectures, the main point of this is to tell you about coordinating different cell behaviors. And so here you have an epithelial behavior, which is this radial intercalation. And you also have a kind of pinching of some of the cells, so that it drives these radial intercalation inside the embryo. For different animals, so for chick, for example, gastrulation happens in a slightly different way. And so what you're looking at here is the chick embryo. And this is maybe around 4 hours after the egg is laid, and the embryo looks like this. And here, this is maybe about 8 to 10 hours after laying. And what you can see is something called the primitive streak. Okay, at the end of the primitive streak is a structure called Henson's node. Henson's node is the organizer of the chicken embryo. The primitive streak is where the mesoderm is being generated. And so here, what you're seeing is these epithelial cells are transforming into mesenchymal cells and crawling away. Okay, here in chick and mouse, I'll show you mouse in a little bit. These epithelial cells form not just mesoderm but also endoderm as well. And so you're getting new endoderm, what's called definitive endoderm, replacing the embryo's old hyperblastic endoderm. Okay, and we'll talk about this in later lectures. But this was to kind of introduce you to another cell behavior that maybe is important to know about. So maybe some of you know what I'm going to say. I mean, if you're reading the slides on the side here, you know that I'm thinking about this transformation of epithelia to these mesodermal cells. Yeah, and what that is called epithelial mesenchymal transition. And so so basically, when the embryo is set up, all the cells are essentially epithelial, but many of those cells will transition to mesenchyme. And so what happens here is that, and so this is what you're looking at here in the streak region. So there is a kind of epithelial reorganization, which is pushing these epithelial cells towards the midline. As they go towards the midline, they're undergoing cell shape changes. The basement membrane, so the substrate on which the epithelial cells rest on, is being degraded. And then some of these cells are down-regulating their adherens junctions, their tight junctions, and leaving the epithelial sheet and migrating away. So this is called epithelial to mesenchymal transition. This is the mode of gastrulation in the chick. It's the mode of gastrulation in us, and in mice as well. And so here, again, you can see the epithelia. So in mouse embryos, the epithelium at this stage is on the inside. And we'll go into this in a subsequent lecture. But again, it's the same kind of concept. The epithelia pushes by rearrangements towards the midline, and within the midline, some of these cells escape the epithelium as mesenchymal cells and then migrate away and populate this interstitial space in between endoderm and ectoderm. Okay? Any questions at this point? Great. So EMT is a particular coordinated cell behavior, which is fantastic. It involves many different kind of changes in molecular configuration. So things like E-cadherin, which is part of the adherens junctions, ZO1, which is part of the zona occludens, the tight junctions, things like laminin, which is important for the interaction with the basement membrane, are downregulated. And as this downregulation happens, you start getting a mesenchymal phenotype. That's shown here. So this is reminiscent of something that's clinically important as well. And so, does anyone want to hazard a guess where EMT becomes very important?
Cancer.
Exactly. Yeah.
Cancer.
Yeah. Exactly. Yeah. So when we're thinking about metastasis, we think about EMT as well. And so these are kind of here. This paper from Jean-Paul Thierry is classing all of the different types of developmental EMTs, but he goes on to talk about EMT as being a primarily clinical of clinical importance, because understanding EMT in development will help you think about EMT in cancer. And as you know, in cancer, EMT is one of the best ways to start thinking about epithelial tumors undergoing metastasis, and then with the reverse process, mesenchymal to epithelial transition, forming secondary tumors somewhere else. Okay, so this is kind of one way of thinking about EMT. The concepts of all of these controlling EMT in cancer have come from developmental studies mainly, and so it's known here. So, for example, here you have it's thought that TGF beta signaling, which is one of a growth factor signaling pathway, is important for controlling epithelial mesenchymal transitions in cancer, and all of this knowledge has come from developmental biology. So development is good for something. Okay, so this kind of this is almost the end of the lecture, but I want you to think about this. So what we've been talking about is kind of this mix of cell behaviors, and so far I've been kind of thinking about this from maybe a single cell level or a few groups of cells, but we want to know how, the main point of development is how do we go from a bag of individual cells to a system with a coordinated set of behaviors deployed in 4 dimensions to enable development. And so this is important. The cell needs to know when to do something, and it needs to behave appropriately. And so we know that cells can sense their environment and react appropriately. So these are all the ways that I could think of that a cell senses some change and does something. I'm sure that all of you will be able to think of a few more. So for example, I think of extrinsic signaling, so growth factors, mitogens, for example, cytokines, hormones, acting on a cell to do something. The cell's contact with the cell matrix, with its matrix, either basal lamina or extracellular matrix. What's in the substrate? What's in the basal lamina? What's around? What molecules are in the ECM? Mechanical stress, nutrition, of course, plays a vital role, and then cell-cell junctions and neighbor relationships also play a role in influencing how the cell reacts and how it behaves. But all of these kind of behaviors are underpinned by gene networks. And so what we're going to talk about in the next couple of lectures is how then gene networks may set up various types of cell behaviors. And so this fantastic review kind of will basically suggest that many of the cell behaviors are encoded by proteins and genes. And these proteins and genes are controlled themselves by different effectors and different upstream regulatory sequences, regulatory components. And so this now gets us to an idea about how cell behaviors can be controlled across the whole embryo. If we can deploy particular signaling molecules and particular gene networks at the right time, at the right place, we can start controlling all of these different cell behaviors to act at the right time and at the right place. And so this coordination of activities does need signaling, because that's the only way that this cell here can talk to this cell here, and so that this cell here knows what the cells around it are doing. And signaling needs to then act on gene networks, which can be deployed in a tissue-specific fashion. So we're going to talk about this in more detail in the next couple of lectures, okay? So I think that's it. So yeah, that's it. If you have any questions, please let me know. Are there any questions? Fantastic. So in that case, can you hear me? I should check that I'm still audible.
Yes.
Yeah.
Okay, thank God. Okay. So as always, this lecture is recorded. I'll put up the slides. I'll write some notes on it as well. I'll give you all a chance to, because some of the concepts I've talked about are kind of tricky, and I'm very sure that I've done a very not a great job of explaining them and trying to convey them to you. So I want you to go away and just think about it a little bit, and then whenever you have a question about it, you can send me a message, or we can organize a session to talk about these kind of cell behaviors as well. Okay, great. So I'm going to use this last 1 minute just to let you know what to expect in terms of evaluations and assignments. So as I mentioned before, I'm only going to give you assignments and open book assignments. And because I am not sure how everyone's connectivity is, and what your kind of personal circumstances are if you're not in Bangalore, sorry, not on campus, the assignment will have a very long deadline. Okay, and so essentially the term paper that Tina and I will set, will have a deadline of something like middle of February, and any assignment that I set for my part of the course will have a deadline of also the middle of February. Okay. So please don't worry about assignments yet. Okay. Cool.
Raj,
Yep.
I had a question.
Yep.
I don't know, it may sound a little vague, but I was wondering, so in the context of going from like a bag of cells to each of those cells becoming some part of the final organism, how much does, so first is, how much does the cell number, I mean, there must be a variation in the number of cells, say embryo to embryo. And then in that context, I'm wondering, say the early cues that come on say, these cells are going to become the posterior half of the organism. How much of a role does the number play on it? Do the cues, are those cues sort of cell specific? Like they pick out, oh, if the cell has this, then I'm going to turn it into like a posterior forming cell? Or is it like a region specific that any cell that forms in say the posterior half of the embryo is going to become this?
So that's I mean that's a really kind of good question, and it's almost verging on philosophical, in fact. So the first part is on cell number. There are mechanisms that exist in every embryo that somehow sense cell number and control for cell number. And in the signaling lecture and Sandeep's lecture, Sandeep Krishna's lecture, they'll become clear what those are. So it's a very it's a very beautiful system. It, involves kind of reaction diffusion essentially. The second part of that question is about patterning, I think. To my mind, the way an embryo is patterned is hierarchical, and so you start off with large regions. So embryo is split up into front and back. And then within the front and back regions, you start getting individual patterning processes happening as well. So between front, you might then pattern forehead, midbrain, etc., etc. And those start subdividing themselves until you do get cell type specific differentiation. But I think up until that point, you're splitting the embryo up into regions, and that regional specification is happening hierarchically. This is shown very beautifully in flies, and so Tina will discuss this in 2 or 3 weeks' time when she talks about maternal effect mutations, gap genes, pair rule genes, and so on. Okay.
Thank you.
Okay. Cool. So if there's no other questions, let's leave it at that. Okay. I'll see you on Wednesday. But if you